So hi, everyone. Thanks for attending. My name is Josh Snyder. This is my colleague, Victor. This talk was also co-authored with another colleague, Laurent Galafassi. I'm going to be presenting the slides, and Victor's going to join me here for questions at the end. And this talk is basically about how we're using Sparc to address some of the database challenges we see in drug discovery. So this is going to be a bit more applications focused, I think, than some of the others. I'm going to give an overview of our use case. I'm going to talk a little bit about the goals of the internal products we built to meet those needs. I'm going to give some insight into the high-level architecture of that product, some of the high-level technology decisions that we made with Sparc and with other components. And I'm going to try to share some perspective on the impact that's had internally at Novartis and also aspects which may be of more general interest. So at the beginning of drug discovery, often there's a process called screening. And screening is basically just scale out of the execution of laboratory experiments. So you can imagine, for example, a single researcher culturing cells or pipetting solutions into a Petri dish. With screening, that Petri dish is miniaturized into hundreds or thousands of tiny wells in a screening plate. And the execution of experiments on hundreds or thousands of those screening plates is automated and carried out by laboratory robotics. So those big yellow things in the upper right-hand corner are actually called big yellow arms. That's what they do. They grab things very precisely and repetitively and move them from instrument to instrument. So this is kind of an assembly line for scientific experiments. And the purpose of doing this really is to find candidate therapeutics. So at the beginning of such a discovery effort, usually there's some prior knowledge about the physiology of a disease. And the question on the table is basically, can we find some compound that has a desirable effect on that physiology, which might therefore be of potential interest in terms of being developed into a therapy. And it's not the only way to answer that question, but one way that that question is commonly answered is by just running as many tests on as many different molecules as possible against that experimental model. So as you can imagine, that produces data, and sometimes larger data. The data size itself is dependent primarily on two factors. One is simply the number of molecules that you screen. And so when I talk about molecules, I'm talking about actual compounds, right? Physical solutions and powders that we have in a library of compounds. This library is maintained and grown over time. It's comprised of extracts of natural products, of compounds that were synthesized in previous research, sometimes of arrays of compounds that are purchased from other companies. And the size of this library can vary from company to company. Some of the larger published examples of library sizes are in the couple of million compound range. So that's a relatively fixed limit on the size of the data that come out of this. That can grow over time, but that's never going to be a geometric progression. We're sort of mostly going to be in that range. The other factor is kind of the technology that's used, and that's highly dependent on the particular experiment that's being run, the experimental design. So screening itself is just a process. It's very technology agnostic. At the end of a screen, you can use high-resolution cell microscopy. You can use different kinds of gene sequencing technologies, different kinds of cytometry technologies, and depending on which of these kinds of technologies you use, you're going to either make many more or many less observations, and you're going to be measuring many more or many less different features for each of those observations. So with microscopy as an example, you might be taking pictures and measuring some aggregate value across an entire cell culture, or you might be taking pictures and segmenting out individual cells and taking measurements for each of those cell objects within the picture. With gene sequencing, you might be taking a few dozen measurements of different expression levels of different genes. And back with imaging, even if you're taking a picture of an individual cell, you might be then measuring a few hundred or even a few thousand different shape or size or image texture-based metrics on those. So the sizes can vary dramatically depending on the technology used and depending on the experiment. But the structure of the data is more or less fixed, and it's very generic. It's observations and features, right? So data tables with observations as rows and features as columns. And the data sets can be fairly large. So I mentioned a couple million compounds maybe. So one screen One full deck screen might be looking at a few million compounds. For each one of those compounds, you might be looking at a culture of a few thousand cells. And in this imaging example, for each one of those cells, you might be measuring a few hundred or a few thousand metrics. And that puts us, that's kind of our upper bound on what we're doing today in terms of the application that we built, and that puts us in the tens of trillions of data points to tens of terabyte range, which maybe isn't extremely massive, but the other point I want to make is just that it's very hard to predict how these data sizes are going to change over time. So again, it's very technology dependent. The pace of scientific innovation as a whole drives sometimes very rapid innovation in terms of the technologies that are used to do this kind of research. So one example probably everyone is familiar with is the sort of super Moore's law cost reduction of next-gen sequencing over the past several years. Even with something like microscopy, where you might imagine fairly hard constraints based on physics and optical limits, we still see year by year instruments coming on the market that are capable of faster acquisitions or higher resolutions. And just to take one example, if you use such an instrument maybe to measure over time what's happening with a living cell culture rather than a fixed cell culture, then you have this additional time point dimension, and even 10 time points in a time series is a factor of 10 on the overall data size. And too many more factors of 10, and all of a sudden we have a much different problem. So that's one of the things that we kept in mind in terms of trying to design a system to handle the data analysis for these data. And that's really the next set of questions, is what do we need to do with these data? So the primary purpose of screen, again, is to find the compounds that show some desired effect. So essentially, we're looking to classify those samples that are active with respect to some control condition. So the example on the screen here on the left, these are images of cell cultures from individual wells from the screening plates. And in the middle are zoomed-in views of individual cells that were detected from those larger images. And the green, in this case, represents a particular protein. So a particular protein that was tagged to fluoresce green. So we actually see green in the images is where that protein is physically with respect to the cells. And so on the top, that's a neutral control condition where you see most of that protein is kind of hanging out in the cytoplasm of the cell. And on the bottom is the active condition where you see it's mostly moved into the nucleus. So that's very easy to see, kind of very obvious when you look at the pictures, but it's not necessarily clear how that can best be quantified. And I mentioned before that we can take sort of tens, hundreds, or even thousands of feature measurements for these images. And that's not really the purpose of our application. We can sort of take off of the shelf computer vision algorithm pipelines to do the image analysis, but then we have this kind of array of choices as to what do we actually want to measure and what's going to best quantify the activity that we're looking for. And in this case, it might seem kind of intuitive that you would look for maybe like the average intensity of green in the cytoplasm versus in the nucleus. You know, maybe take that ratio. That seems like probably a pretty good idea. And that is a pretty good idea, but it's not the best idea. It turns out that something called cell texture variance 3 pixel actually gives you a better separation between those two phenotypes. So the ZPrime number on the bottom, that's a statistic which indicates sort of the goodness of separation between your active control on average versus your neutral control on average with respect to that particular metric. So the higher ZPrime number, that sort of better differentiates those two phenotypes. And that would never be immediately obvious, right? Maybe you think, so cell texture is, in retrospect, cell texture is kind of measuring, image texture is kind of measuring the smoothness of the image. Sort of, is it smooth? Is it dappled? Maybe are there hard lines? And you could go back and look at the picture and say, okay, that kind of makes sense. I could see how that makes sense. But you'd never know that up front. And you'd certainly never know whether the 3 pixel version or the 5 pixel version is going to give you a better Z prime. And for me, this is kind of the metaphor for this problem is Flatland. So Flatland is this sort of classic book that has a thought experiment about a two-dimensional world that's just a flat plane with two-dimensional inhabitants. And what would happen if they were visited by a three-dimensional sphere? What would that look like to them? Well, they can only ever see one slice of that sphere at a time, right? It looks like maybe a circle to them, or maybe a point, depending on where it's intersecting with the plane. So depending on which slice they look at, they're going to get a better or worse sort of description of what's really there. And if they could actually look at all of the slices, they'd get an even more accurate picture. And so that's kind of what we're trying to achieve here, right? We can only see what we look at, and so clearly we need to look at everything. We can't just limit ourselves to looking at a single feature. And today, sort of that's the standard in a lot of this kind of research. So there was a meta-analysis done recently of literature of imaging screening data that was published, and I'm probably going to misquote this, but I think it was something like 60 to 80% of all published versions of this kind of research are either only looking at a single feature or completely ignoring that cell-level data and just looking at sort of aggregate measures across those cell cultures. So we really need to look at everything to get the best answers. And in terms of looking at everything, as I mentioned before, the structure of the data is very process-driven, and so is the analysis, in fact. So in the case of imaging, of course, there's some pre-processing to get the feature metrics. You get different kinds of feature metrics, whether you're doing imaging or doing gene sequencing. But at the end of the day, you have the observations and you have the feature metrics. And you need to do pretty much the same kind of series of analysis pipelines, no matter what technology is used to produce the data. So you need to find, maybe if you're looking at multiple features, you need to find features that are highly correlated with each other, or features which have very little variance, so they aren't really informing anything, they aren't contributing to the analysis. You actually want to get rid of some of those features. You want to be able to look across features and do kind of classification based on full or maybe reduced feature metrics. There are different kinds of QC measurements you want to take, aggregations you want to take across observations, different kinds of normalizations and scoring and kind of artifact correction you want to do. And that's essentially the analysis pipeline, or rather series of analysis pipelines that we wanted to build. And so that kind of smells like Sparc. I mean, each one of those is really an independent data pipeline, and it has a number of individual steps, but it doesn't really require interaction inside of it. It does require interaction, visualization, and decision-making in between each of those stages. So kind of taking this level of abstraction above, this is basically a series of batch data pipelines. We have some visualization and control and interaction that we want to do in between. And the kinds of transformations and computations we want to do are fairly standard and very well suited for Sparc. It's aggregations across rows, across columns, different kinds of transformations. So we thought Sparc would be a great fit. But Sparc is not really a great tool for the biologists and the screening scientists that are running the screens. We wanted to get away from this world where every time you run a screen and you have a big data analysis to do, we need some kind of specialized, bespoke, custom-coded, and even custom-executed version of a data pipeline that someone has to run for the screening scientist. So we wanted to build an application around these data pipelines. And that means we need the visualization and control to be done in sort of a very friendly and constrained manner. And we also have a whole layer of workflow logic that we need to put in as well. And that's kind of what drove our really, really high-level architectural decisions. As I mentioned, Sparc seemed like a great fit for the data pipelines. Obviously, due to the data sizes, we need to distribute the computation. We need to make sure we can run it efficiently. The declarative API makes it easy for us as application builders to implement the methods for the data pipelines very quickly and implement new methods much more quickly over time than if we always have to worry about the details of distributing the data, distributing the computation. R and Python support was kind of icing on the cake. We also have more of the analysts and informatics users. And the overall ecosystem was ended up being really useful as well. MLlib obviously had methods that we could reuse. Sparc Job Server made it easy to integrate with the front-end layer. And in terms of the front end and the visualization layer, again, we wanted to sort of actually make it more constrained here. We didn't just want a window onto the data, sort of a very thin window. We wanted to have sort of very specific visualizations, but also very demanding visualizations. So for that, we wanted to build a web UI that also let us constrain the workflow and make it very simple and linear. And we used WebGL so that we could actually render sort of a fairly large number of points in real time interactively in that web UI. And so I'll show a few examples of what that looks like in a minute. The final sort of big piece architecturally was Cassandra. To drive those visualizations, we wanted pretty fast random access to different chunks of the data. And Cassandra was a good choice for that, not least because of its Sparc connector. So this is just one screen from the application, and the main point here is that, again, we tried to keep it simple from a workflow perspective. Along the top, you have the tabs. It's very linear. You go from data input to that kind of QC, the normalization and the scoring, and finally that classification step, that hit analysis, where you finally determine which samples actually kind of look like they're active with respect to the control conditions. And then for each one of those tabs, we have this kind of panel of options, a bunch of UI controls which are specific to that data pipeline and which parameterize the execution of that data pipeline. And then it's just a run button at the bottom, right? We click that button, that's a Sparc job server message, that goes to our Sparc cluster and executes that data pipeline. Different kinds of computations take different amounts of time, so some of these are, and also depends on the data size, right? So some of these are almost interactive, on the order of seconds. Some of them are still on the order of hours for our current pipelines. And we also wanted this kind of interactive visualizations. Okay, yeah. So that's WebGL for us. That's what it gives us. I mean, this is only 50 plates, but we can do this with a few million data points, which would be like all of the few million samples that we might be screening, right? So it can give this sort of broad view over all of the data with respect to a particular feature in this example. And it easily lets you change the heat scale and maybe pick out individual plates of interest, things which are outliers or which show artifacts, which you can then choose to exclude from the analysis or mask out of the analysis. And so we did kind of do a bunch of very standard visualizations, heat maps, histograms, bar charts, but we did custom implementations with WebGL to kind of enable this level of interactivity. And sort of the final piece I wanted to tell you about were the actual algorithms implementations. We managed to reuse some things from MLlib, certainly some of the more finely grained methods. In terms of classification, we kind of rolled our own. We did an implementation of Mahalanobis distance. We did a Gaussian version of naive Bayes. And we rolled a bunch of our own kind of either more coarsely or more finely grained utilities. So for the multivariate analysis, we did our own fine linear combinations method. Some of these are kind of mimicking things that we were sort of more familiar with doing out of the box from like our Caret library. And some of them were even sort of just new and very specific to statistics that are relevant for screening. Imputation, different imputation methods in case we have missing values or different ways of handling missing values, case-wise versus pairwise. This wasn't always available, obviously, out of the box from MLlib. And so for us, the big picture really is that we built this application that works. So we have a version 1 out. We were able to kind of abstract the problems of analyzing all these different kinds of data coming out of different technologies from different screening campaigns. And before we did that, the model was very much, well, there's sort of an analyst, an expert in a particular area, maybe even on a particular technology or particular kind of data, who is responsible not only for coding the analytical pipeline, but also for executing it. And now we've abstracted that so that it's, there are always edge cases, but it's more or less common across a number of these technologies. It's kind of the 80-20 rule. We can satisfy like 80% of the use cases this way and have a very reusable and repeatable tool and a repeatable way of doing these analyses. It also scales with data size. And just in terms of execution, in terms of that scalability, I mean, we're running certain data sets of certain sizes today. And we have a cluster of a fixed size today. But what we've seen just practically is that we can sort of reduce these multi-day batch jobs, what we saw as multi-day batch jobs before, into maybe multi-hour or just multi-minute jobs now. In terms of future work, we want to make it elastic. It's very spiky what we get from these screens. Sometimes we get big screens, sometimes little screens, sometimes more screens are happening, sometimes more people want to do more data analyses. So we want to be able to kind of auto-scale in response to that demand. We want to implement more methods. In terms of that individual cell level analysis, we want to be able to detect phenotypes that we may not expect based on these large feature vectors. We want to be able to give user input into that, so some more supervised learning methods. And ultimately, we want to expose the APIs for the methods that we do implement internally to our informaticians so that 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 10 or 20% use case where they still have to do work, they can still focus on that, but they can leverage Sparc and they can leverage the distributed computation and the parallelism. And finally, we hope to kind of refactor some of these to the point where we can contribute them usefully to some open source package. We're not quite there yet, but that's one of our next goals. And the really big picture, I think, is that now we can kind of draw this line from Sparc to, actually having an impact on accelerating the discovery of new therapies for patients in need. And for me, really kind of looking across the industry, there's like this massive pace of innovation in the technology industry as a whole, and all these great new tools coming out and being used in amazing new ways, right? That doesn't often happen in healthcare research. I mentioned pace of innovation, I mean next-gen sequencing, stuff like that happens, but the use of information technology to really enable and accelerate healthcare research to date hasn't really happened that much. And to me, this was a really exciting example of finding a really good overlap between one of these tools coming from the broader industry and being able to use it and really achieve something in drug discovery research. And there are a ton of people that contributed to this, not just Victor and Laurent and I, but from requirements to implementation and actually many, many more than I can list on this slide. So definitely thank you to everyone who contributed to this. And thank you to all of you for coming and listening.
This is a cool application. Could you comment about the WebGL portion? Did you write your own shaders, and how did you do the aggregation for the WebGL data?
Yeah, so basically, we're sort of doing some pre-aggregation, right? So the aggregation is still happening in Sparc, and depending on what we're trying to visualize, we're either visualizing aggregate data or some chunk of the original data, right? So for those heat maps views, it's maybe the raw data in one case, or maybe it's the scored data in one case, but it's still like one data point per one of those original feature observation data points. So that's actually not sort of aggregated at all. That's sort of already constrained in terms of looking at data for a single feature. And you can imagine when we get to looking at individual cell population levels, we're going to have to do a similar kind of constraint. If we want to look at a cell population, we're not going to look at every data point for every cell, for every well in the entire data set at once. That will be too much for WebGL also, right? So in that respect, sometimes we do the aggregation as part of our data pipeline in Sparc, and we pass the results back in basically just a kind of data structure that goes back to our front end. And yes, I actually personally didn't do the WebGL implementation, but yeah, I think we wrote our own shaders, or the guy who did that wrote his own shaders. Did that answer your question? Okay.
Please tell me about the infrastructure, like which, is it on-premise bare metal servers or public clouds, or how many servers do you use?
Yeah, so I'll mostly hand that question over to Victor, but today it's on-premise, but we are moving to the cloud. That's also just more of an organizational readiness question, but we want to get it into the cloud, and we want to get it very elastic from that point of view. For more details, I think Victor can speak to that.
So right now we have 8 worker nodes, a cluster inside the company, in the data center, and on the virtual machines. So 8 nodes for worker, 3 nodes for master, and start out with 32 gig of memory. Now we're running 64 gig of memory on those virtual nodes.
And I should quickly mention that's not really for that like 10 terabyte use case. That's more for the use case where with imaging data, it's already aggregated at the well level.
Private cloud, yeah, in our own data center. But we are in the process of moving into the public cloud, Amazon, for example.
So how long did it take to write the application and put it in production?
So version 1 into production was roughly 8 or 9 months. and we're now executing on version 2, the new set of requirements, new directions we want to take. So some things are just sort of hardening that first version, but other things like the elasticity and the supervised learning of phenotypes and applications of that to sort of treat independent cell populations independently is much bigger in scope and may even not fit into sort of our next phase. We try to time box it into sort of 6-month or so delivery cycles.
How do you integrate Sparc workflow and, say, R?
So that's also future work, basically. We have a lot of pre-existing algorithms implementations from informaticians within the company that are mostly in R, actually. Sometimes Python, sometimes something else, but often in R. And that was one of the attractive features of Sparc for us when we were just evaluating at the beginning what we wanted to do. But we're not quite there yet. And we're still in version 1.3 of Sparc, so we're now moving to the latest thing. And I think even at that time, the R support was a little bit less mature. But we want to get to the point where it's easy for informaticians to write algorithms and sort of dump them into one of our data pipelines, maybe with a little bit of porting or a little bit of additional development. But we want to reduce those sort of barriers between what the informaticians are doing and what we can sort of make a little bit more reusable and consistent in our application.
We should do a poll. How many pharma or biotech companies interested in doing something like this or thinking about?
Well, how many are in the audience in the first place? Maybe not many, I don't know.
Quick question. You said this is high dimensions, and you mentioned there are a lot of dimensions. You're using Cassandra to store this data. Can you give a little bit of idea on what's really happening in that part?
Yeah, I'll let Victor take that one too.
So each dimension is, in a Cassandra is stored as a cluster row, right? So a partition row. So you can go as many rows as you want. So we initially designed to store it for 500 features. We actually have customer run for 2,000 features. In terms of each feature, we have designed for up to 1,000 plates, and these plates becomes a column in the Cassandra, and then within each plate we have 1,500 values are packed in a blob. So that's roughly. Is that helpful? Yeah. So we're for the size we have right now, we're pleased with the performance of Cassandra right now. But of course, we're going to increase the size of the data in the future phase. So we'll have to see.
Hey, I'm just curious if Sparc has changed the way you design experiments at the bench level.
The user design experience?
No, just the way you design experiments.
Oh. The way we design experience.
Experiments.
Scientists
design the
I would say definitely not. We're not trying to drive the science in any particular direction. I think it's changed the way that people are approaching their data, and I think it's still a bit early days for us from deployment, but the more that the scientists use the tool and the more that they have the capability of dealing with sort of all of the data, all of the measurements, it's going to change the way they analyze their data. I don't know that long term it might change the design a little bit, but that might be a slower process, right? But definitely, like I mentioned before, right? People are doing these screens and they're saying, all right, I'm going to run this thing, and I'm going to get 500 features for my image analysis, and I'm going to throw away all but one of them. And I'm just going to assume that one is going to help me find the samples that are hits. That's changing, right? That's changing very quickly.
One thing, though, I think it does help them to think about using more fancier instrument. Like they have a fancy instrument, but they're not using all the features of it. So now they have the ability to handle the data. They can start using them. Yeah.
That's right. Also, right. they get these, you spend maybe a few $1 million on an instrument, but there's still this mindset where maybe people aren't even thinking about what they're going to have to buy in terms of storage to like deal with the data that are coming off of that instrument. So it's not even at the level of Sparc and data analysis. We're also just even, I would say, like just planning within the organization more broadly is sort of becoming more aware of this, mostly just because of the data sizes are going much too big, much more quickly than we can handle. So just the data pressure alone is kind of causing people to rethink and causing the business, right? It's ultimately the scientists who are kind of championing this, right? They're the ones saying, well, we have these data, we can't deal with them, we're not using our instruments to the full capacity, so please help us. And that's what was exciting about this, was Sparc kind of gave us a way that let us do that. I mean, 9 months sounds like a long time. To believe me, in pharma IT project, that's that's like a sprint or something. But yeah, so.
Are you using ML at all, and if you are, what components?
Well, we're using the correlation matrix computation part right now. The algorithm we actually want to use is not in the ML lab right now. So hence, in the near future, hopefully we'll help to contribute to that. I think
we use some smaller components like the correlation matrix. We do a lot of the local math using Breeze also in our Scala implementations. Yeah, but some of the really the high-level algorithms, the more coarsely grained algorithms, we didn't find a perfect one-to-one fit yet. So like the Gaussian naive Bayes, for example.
Any other questions? All right, let's give them a round of applause.